Welcome children. Today once again we are going to discuss about practicals in biology section. So in our previous session we studied about one of the practical in which we studied plant cell in which the sample was taken from onion peel. So we studied about the various procedures and precautions, observation, interference, etc. And in today's session, we are going to discuss some of the questions which are related to that practical only. Viva is a very important part of practical examination. So in today's session, we are going to discuss some of the questions which can be generally asked in Viva in the form of assessment. So this time, if we are not able to conduct the practical exam, at least we should practice some of the questions which are taken from practical so that we can give such kind of question in your actual theoretical exam also. So don't take it lightly, children. Pay little extra attention towards these practical questions. So the first question is, name the type of microscope used to observe the slide of animal or plant cell. The answer to this question is, one should use compound microscope with lenses having the magnification of 10x, 40x and 100x. There are gradations in magnification which starts from 10x, and if you want more magnification you will have to adjust it on to 40x, and then further magnification is available. Then the next question is, why do we use a stain for mounting a slide? The answer to this question is very easy. Stain makes our visualization very easy. When something is colored, you can very easily observe that structure. So the stain makes cell wall, nucleus and cell membrane visible as they are colored or they take up the stain. The next question is, why can't we see all the cell organelles under microscope? Why we can see only cell membrane or cell wall and nucleus and cytoplasm? So the answer to this question is, the other cell organelles do not take up the stain and are too small to be seen under the lab microscope. They are very small structures, so we cannot see it under lab microscope. We need to have electron microscopes to visualize such kind of structures which are very much smaller in size. Now the next question is, give the use of glycerine in slide preparation. Glycerine prevents drying of specimen. Children, you must have noticed at home also during winter you apply glycerine onto your skin so that your skin will not be dried up and it will remain hydrated. So what happens here? Why do you use glycerine on your skin? Glycerine makes a kind of a layer onto your skin so that the moisture content will not go through this layer across your skin. The same thing happens here. When glycerine is placed over your specimen, the specimen will not lose its moisture content from it. So it prevents the drying of specimen. Now the next question is, how many types of lenses are present in a compound microscope? There are basically two types of lenses which are present in a compound microscope. The first one is present in the form of objective lens, and the second one is present in eyepiece. So there are two different types of lenses in a compound microscope. Now the next question is, give one example of epithelial cell. There are so many examples of epithelial cells which we have already studied during our explanation sessions. But when we are talking about this epithelial cells in practical, especially when you are making a slide with a cheek cell, then your first spontaneous answer should be cheek cell. So cheek cells are a kind of a epithelial cells. Now there are some practical based questions. So let us discuss these questions also. The first one is, what should be the lens magnification when you place the slide under microscope? The answer to this question is, initially the lens with a lower magnification power, that is 10x, should be used to see the material, and for viewing under higher powers we use higher magnification lenses. The next question is, what is the position and location of nucleus in animal cell and a plant cell? So answer to this question is, in plant cell nucleus is present on one side of the cell, whereas in the animal cell the nucleus is present in the middle of the cell. Now the next question is related to the previous question only, that is, why is nucleus present on one side of the plant cell? So the answer to this question is, the plant cell consists of big vacuoles which occupies the maximum space in the cell, hence the nucleus is present on to the side. The next question is, what is the function of cover slip in slide mounting? A cover slip keeps the slide mounted material at a fixed position on that slide. Otherwise, it may be possible that when you are shifting your slide from microscope to your workstation or slab, or from one place to another place, your mounted material can slip away from your slide. So to fix that material onto that slide, you need to cover it with a cover slip. And this covering prevents the material from drying also. Not only this, there is one more function of this covering of material with a cover slip, that is, it also protects the delicate and costly lens of the microscope from getting in contact with the material, which can lead to fungal growth onto the lens. So it ultimately protects the lens. Now there is one diagram based question. In this you will have 4 options. You have to choose the most appropriate correct option out of these 4 only. So look at all the options one by one, then give your answer. The first option is this, in which cell membrane is present, cell wall is shown inside, which is a wrong position actually. So this diagram is wrong. Here in the second diagram, B diagram, you can see nucleus is properly denoted, vacuole is correctly marked, and cell wall is the outermost covering, and below that cell membrane is there. So this one is the correct answer. Now moving on to the C option, that is showing nucleus in the center but cell membrane inside the cell, which is a wrong answer. Now here in this D option, you see cell wall is there, cell membrane is correctly placed, but there is interchange in the labeling of nucleus and vacuole. So this is again a wrong option. So the correct answer is diagram number B, in which all the parts are correctly marked. Now move on to the next question. Which one of the following is absent in animal cells? Cell membrane, nucleolus, cell wall, or cytoplasm? The answer is very easy, cell wall. Now pick the odd one out. There are 4 options. The first one is plastids, second one is large vacuoles, third one is cell wall, and the fourth one is centrioles. So the answer is very clearly visible here, that is centrioles. Centrioles are present only in animal cells, not in plant cell. And rest other organelles, plastids, large vacuoles, and cell walls are present in plant cells. So here the odd answer is centrioles only. Now the next question is, name the stained part of the cell. In onion peel, the parts of the cells stained are cell wall, nucleus and cytoplasm. Thank you.